Welcome to practical 1 in our course G07 on phylogenomics. So we talked to the introductory lectures, and we're going to use some of these methods in this lab, including constructing basic trees and trying to look at different substitution models and different data sets and see how their phylogenies are constructed. So what we're going to do is firstly install the relevant packages that we need. We're going to create a tree with a small set of 7 FNDV VP1 sequences, where you're going to use a package called ape to make a phylogeny from those sequences. We're then going to create a large-scale FNDV VP1 tree. We're going to create co-phylogenies that represent different patterns of trees for different sequences for the same set of samples, so that we can compare them against each other to see if patterns of ancestry differ between those two trees. And then lab 2 will look at linking metadata to large-scale phylogenies, looking at DNDS ratios and how we can calculate them using R. And then we'll move to the server to use model test ng to select a substitution model and construct a tree with RaxML. So that's our kind of goals here. We're going to talk through each stage first on the slides before going to do it on RStudio, the server, as well as relevant. So what we're going to do first is install these packages via Bioconductor, BioC Manager. We're going to use genome info DB, which is a required package for biostrings and needs specific updating in my experience. We'll use MSA for constructing multiple sequence alignments. We'll use Sequenor for obtaining sequence data and ape for phylogeny construction. I'll then use these accessions as an example set of samples for foot-and-mouth disease virus, the VP1 region only, construct a basic tree based on those short sequences. What we'll do is use a function in read.genbank from Sequenor. We'll take that list 1. We set our sequence names to be list 1. We want the species name here, and we're treating it as character. So we can record our sequences in this variable whose structure we can examine next. We can look at the attributes just to see what's inside there for the species attribute, and look at that further, basically. So we'll see something like this where we look at the sequences and their corresponding sequences. So they're all FMDV SAT2, and then we'll be able to write out our downloaded data using write.dna. We'll write out the sequences to a file. It's in the format of FASTA, where appending is false. The number of bases per column and for width is just cosmetic. It doesn't matter too much. And what you can do then is use this code to get the MBank accessions of your own and download them. So let's look at that now on RStudio. So if you don't have BioC Manager installed, you can do that. I have it installed, so this should evaluate as not being required. So that's already installed there. I have Sequenor already installed, so if I check that with the library Sequenor, it appears like this. Ape, again, I have that installed. I check what packages are inside ape by typing in ape and then 2 colons, pressing tab, and that gives me a list of files then that appear that I can use. We can install genome info DB as well, which I've installed already. MSA, again, when you install the library, you should see nothing appearing. So if you see a request to update other packages, do so and hit A for all. You can set your working directory, again, using session set working directory. You can go to source file location is probably the easiest. It depends on how you set up your file folder. What I want to do then is take these accessions we have listed here, and what we're going to do is record them as a list. So this is the structure of list 1. It contains some short sequences. We've got 7 entries inside there, and we can look at list 1, entry 2, for example, by just getting that elements 1 to 7 that we have. And then we use the function read.genbank, so we can check that function by doing question mark and that function name. We see it appear on the right, so that's in the language with the package ape, not Sequenor, it turns out. And what we're giving it is that we want to extract these sequences in essence in containing a list 1 from GenBank. And we can check in seeks what's in seeks with an S. So there we see 7 attributes for the species, and these are the actual sequences that we have there. and that's present for each particular sample. So if we look at seeks and then do dollar, we see we have each sample there, and it's actually, you can extract each site from the sample if we want to do so. We can do that all together. So what do we download? We download those sequences. They're 648 base pairs. Get the attributes of the species, check the species, and check that they're all FMD SAT2, because if we accidentally download something else, that would not be useful. So we can write out those files and then check that they're downloaded on our local computer. And now we'll move on to aligning the files in the next portion. So we'll go back to the slides. So what we're doing is taking our sequences.fasta file that we wrote out. We're going to read it in as a DNA string set. So it's a set of characters. We're going to align it using muscle. Plus Omega is also available. This is using the package MSA and its function MSA to align the sequences. So when that's completed, we can use this print command to print the full alignment, and we'll see something like that. So you can see it's highly conserved bar the odd site that varies, meaning that it's a nice set of sequences just to align and assess the mutations that occur at different sites, and use those mutations to construct a phylogeny as we go along. So you can see, yeah, there's certain SNPs that are present, but then also some conserved regions as well. So next we can align them using the MSA convert function, taking sequence lines that we had, and we're calling it a sequenor alignment. We can then construct the distances for the alignment, create an identity matrix from IA, which we created before. So that's seek.distance that we created. We can view that matrix, and what we see is the pairwise distances between the different cells is based on the number of fraction of nucleotide changes. So you see that some are more divergent than others, and it varies a bit. We then use the plot.philo function to create instruction to visualize a neighbor joining tree, the NJ function here on seek.difference. So we're putting the distance matrix into the NJ function to create a phylogeny of the FMDP VP1 region. We're adding a root, and we're calling it a phylogram here, and we're turning off the tip labels for simplicity here. We can use this function called locator to give us the coordinates for X, Y coordinates. So we clicked on the plot when we do this function. When you run this line, click on the plot twice, because we got locator 2, so 2 for twice. And then we just configure it then X1 is equal to 0.28, and X2, the other Y coordinate, is 0.9. That means when we add the scale bar, we can identify these coordinates more easily. And that's the number of substitutions per site for the length that's shown. So what we should see then is something like this at the end. So we can see there we have the names for the different samples. The substitution per site is down here at the bottom. And we can see their similarities. So they're quite, there's moderate diversity present. So let's look at how we can do that with R. So what we can use is this tool, read DNA string set, this function, and we're sending that to MSA, which from the MSA package. And that's in the biostrings class, hence that's why we need that package. So align that takes a very short time because we're just using short sequences. If we print out the alignments, you can see there, this is the final basis there for the 7 samples. And you can see there's moderate conservation, but certain nucleotides do change. We can then create a variable called myA, which contains that sequence alignment in a kind of computer readable format. So you can sort of see it there. There's only just a few gaps inserted. We can then create our distance matrix and view that matrix, which we can see there. The round function just means we're only showing 3 digits. Let's remove that. And then we have NJ seek distance. That creates an NJ variable from there. So we're sending that into plot.philo to visualize it. So that's from ape, and that visualizes plots. So we can call it as plot, but that might conflict with the inbuilt base function plot. So that's why I use plot.philo just for clarity. So we might start with an initial plot like this, where we just have no parameters. So let's move this over a bit. But then we may want to add on things like the root.edge function. So you can see the difference there. We're creating it as a phylogram by default. We're adding the title in there as well. And then we're saying show tip label is true, which it is by default, but it's nice to know that you can toggle that on and off like this. And then we can add the scale bar by just identifying the coordinates that are best to use. And here, X1, Y1, we're going to use them, and we're going to 0.05, a shorter scale bar. That didn't work. That's because I moved the, let's try that again. So we'll put it here. So that means X is about 0.23, 0.24 perhaps. And Y is going to be 1.05. Okay. That's better. So let's try that all again. So that takes some effort to get right. As you can see, each time you change the plot size, the coordinates will change slightly. So that's why the locator function is handy for some trial and error. So next, let's look at using ape to make a phylogeny. So at this point here, we're using ape to make a phylogeny. So what we're going to do is a more flexible approach. What we're going to do is input our sequence.fasta, and we're going to read that as a DNA file like before. We're going to input that as a matrix using a different substitution model. So now we're going to model using character-based model the sequences that are present in the TN93 model. You can change that to a different substitution models, the JC69, K80, F81, K81, F84, T92. You can also toggle gamma to be true or false, though generally speaking, it's probably better to leave it as true. We're going to put those DNA distances into input by 2 and construct a tree from that. So we're going to use a variation of the NJ algorithm known as BioNJS, or the function BioNJS that comes from the BioNJ algorithm. This can tolerate missing data. So we put that into input 3 that we can send down here to plot. So here we can create our own rooted plot. So you can see the relative similarity. We have sequences 4, 6, 5 clustered together in 2 and 3, and then 7 and 8 are down here. We can create unrooted trees as well. This is a cladogram, which is not ideal. We can create a radial type tree as well, which you see here again. That doesn't work for this particular sample because there's too few of them. So probably the best option is a phylogram like that. So again, we see 5 and 6 together with 4 somewhat related, 2 and 3 together, and 7 and 8 together. And then we use the locator function then to add on a scale bar like the one shown here. So let's look at this then on RStudio. So let's read in our DNA sequences into input 1. We can check input 1 here like that. So we see here our sequences have been read in like before. We use this TN93 model. Let's try F84 here. Oh, gamma correction is not available. So what we do is toggle that to false. And then input 2 contains those sequences, and it's a pairwise distance matrix. So if you view input 2, we'll see a pairwise matrix of distances between the different samples. If we try different substitution models, we'll see that they vary slightly depending on what approaches they take. And then we can use BioNJ to construct a tree from that. But like above, this tree is not yet visualized, and hence we need to use plot.phylog to visualize that effectively. So we can create an unrooted tree like that. And we have the radial version here. And then we have the option to root the tree using the root function. And we can create a phylogram from that. So you see here we have 7, 8 close together, and 2 and 3 close together, and then 4, 5, and 6 are somewhere closer together. We look at the previous trees. We can see that we had a similar pattern before, but at least now the names fit inside the plot, which is useful. And then again, we can use the locator function to see if we can place the scale bar appropriately. Yeah, it sort of fits on there. So let's move on next to a different part. So now we're going to make a large scale tree from all available 99 FMVV BP1 sequences. So what we're going to do here is use a package from Entree to help us retrieve those sequences from databases. So we use Entree search from Entree. We're using the nucleotide database. This is our search term, like if we typed it into a website, FMDV virus and BP1 gene, returning a max of 99 sequences. So if we request too many sequences, we may get an error in downloading them. So if we want to collate many sequences together, we have to slowly add them together and append that to a single file. So we're getting these for VP1 gene, and we have to check how many did we get. So we can use our search IDs then to retrieve these from the nucleotide database. So it's a search term to get the matching accessions. Entree-Fetch will retrieve those exact sequences. We're getting them in faster format, we're storing them in sequences 5, and we can write out sequences 5 to our local folder for safekeeping. In the same way as we did before, we can read that in as a DNA string set and align them, bearing in mind that it will take longer if you have more sequences. This time we're using also close to omega, just so you can see that there's different options there for alignment. We can then convert that into a distance matrix and align that using plot.philo again, this time using a bit radial tree. And then if we do that, we'll get something like the plot that's shown next, something like this. We can create a phylogram as well, which maybe is I don't know which is better, it depends on your preferences, but that also is effective. So let's try doing that on our studio now. So we have to install Entree first before we do all this, and that's installed here already, which is convenient. And what we can do then is, I'll just widen this so that we can achieve that search. So we search the term foot disease virus and PP1 gene, returning maximum 99 sequences. If we look at the structure of that, we see we have a bunch of accessions essentially in there. If we make a error, so if we search for that term, we'll find that we get nothing. So it has to be the correct match. If we make it too general and not specific enough, not specific for a gene, we might get a mix of different things which you don't want. So we need some sufficient specificity to get what we want, which sometimes does take some practice. So you have to bear in mind that for the 99 we've selected, the label is fundamental disease VP1, but that doesn't mean that they're actually correct. So those are IDs that we have. We use Entree-Fetch then, so we send those to Entree-Fetch to get our sequences. We look at the sequences, we can write them out to file. Sequences 5, you can see them all mixed in there. And you can do that for your own organism if you want to do that as well. So as before, we can take MSA and read in those sequences, align them with cluster omega. This will take a little bit longer because there's 99 of them. We can convert them into a distance matrix and plot them using the naming joining algorithm as before. Let's try to make it bigger. If we turn show type labels to be true, you can see that it's not as easy to see. So that's why that's set to false, unless you want to set the text size to be very small. That doesn't really help. We can add a scale bar as well. So you can see the scale bar. A phylogram is probably most effective here because that radial tree, you'd have to send it to file to get it visualized properly. And we can add a scale bar on here. So it sort of works. If we show the type label to be true, you can see that it's not very visible. We'd have to turn the text size to be very small to get it to be more effective. So now let's look further at making co-phylogenies from making just single phylogeny. So now we're going to look at making co-phylogenies as the end part of our lab one. So here what we want to do is visualize two trees and link the samples to each other. So here in this particular example, you see a lot of heterogeneity between the two different alignments on either side, because we have 7, 8 placed down here, they group up here, but they're in a different arrangement. 2 and 3 are somewhat more related, but down here they're at a different configuration. So in truth, these two trees are somewhat more similar to each other. It's just that there's some subtle differences that mean the configuration is topologies are quite different. So we're going to use this phi tools, so you can install that first. We're going to create a distance.dna tree as well. We're going to use a model of G07, but you can change that. We're going to create a tree using the BioNGS model. And then we're going to use the copilot function where we put in the two inputs we have. So our old tree and our new tree model with a different substitution model to see what difference it makes. We can repeat this and try different models. So we look at K80 model as well and compare that to original tree, which was F84 in the particle here, and compare that to the K80. I see that we have good alignment between those two. You see very few differences there at all, virtually none, in fact. And then finally, we can compare that to K81 versus the original. And again, now here we have more differences, but again, it's more subtle in this particular instance. So then let's look at this on our studio to see what's going on. You can see here the key thing is this copilot function, which takes in two trees as arguments with the rotate function as well to rotate the samples to match each other where possible. So we check that we have 5 tools installed. We construct a new mutation model for the same data using G69. Let's TN92 instead in this example. We can construct a distance matrix from that, plot as the cophylogeny between the old data and the new data. So here it's not very visible, so let's try to make it more visible. Let's try that again. There we go. That's more visible. So you can see we have a perfect match, and in truth they're actually identical. So let's try the K80 model versus the original tree. Rotate that. We can see that now we have some cosmetic differences because 2 and 3 are more related to 7 and 8. It's a major difference, I think, in the original tree versus the model with K80. So the substitution model has changed our topology here. If we try K81 and the gamma is not possible for K81. Let's try that again. Again, we get a perfect match. So 2 different models. We had T92 and K81, where we see that they actually come up with the same topologies in this instance. So that's where cophylogenies can be very useful for, say, comparing the phylogenetic signals across different sites, especially for recombination detection. You might have your core region of interest, and you compare that something that is potential recombinant against this core, and representing that as a cophylogeny is an ideal option for that. So please see Lab 2 for more details on how to further develop your skills of visualizing phylogenies.